Ribosome active centers and biogenesis of ribosome in eukaryotes. Introduction. The ribosomes are first noted in plant cell by Robinson and Brown in 1952. During mid-1955, George Emil Pallet observed them in animal cells by electron microscope as dense particles, first named microsomal particles. Its detailed structure was discovered in 2000 and 2001. George Pallet. Cells have tiny granular structures called ribosomes. Ribosomes are assembly shop for protein synthesis or site for protein synthesis, also known as translational apparatus. Ribosomes are present in both prokaryotic and eukaryotic cells. In prokaryotic cells, it freely seen in cytoplasm. In eukaryotic cell, mostly seen attached to rough endoplasmic reticulum and also found free. Non-membranous organelle made up of rRNA and ribosomal proteins. Here you could see a picture of ribosomes. Then ribosomes are made up of proteins and ribonucleic acid in almost equal amounts. It comprises of two sections known as subunits, large and small. The tiny place in smaller subunit in mRNA binds and decodes. The tiny place in smaller subunit in mRNA binds and decodes. The larger subunit is the place where amino acids are included. Small subunit, mRNA. Large subunit, amino acids. They interact with each other. The ribonucleic acid is obtained from the nucleolus at the point where the ribosomes are arranged in a cell. So prokaryotic ribosome 50S and 30S, together 70S. Then eukaryotic ribosome 60S and 40S, forming 80S. Ribosome active centers. There are mainly 4 binding sites for the binding of mRNA and tRNA. In addition to this, there is extra 2 site also can be seen, which is the peptidyl transferase center and translation factor binding site on the large subunit. Then M site. It is the mRNA binding site in the small subunit in the region of groove dividing the head, the body and side bulge of it. 3 proteins called S1, S2 and S5 can be seen in mRNA binding sites. Protein S1 occupy in the interline position on the external surface of small subunit in the region of the neck. It has an ability in forming complex with polynucleotide. Binding of this isolated protein S1 with mRNA results in unfolding of mRNA secondary structure. Next, protein S5 is located in the 30S subunit head near the neck and the S3 subunit of the opposite of that S5. Structure of ribosomes. Ribosome mRNA large subunit. tRNA docking sites EPA. Large subunit modeled 3D1 simplified view is also there. mRNA binding site. Then may enlarge this picture. We could see portions. Polypeptides. Amino acid, ribosome, anticodon, tRNA, mRNA, codon and codons, mRNA binding site, etc. A site, amino acyl site or acceptor site for the binding of incoming amino acyl tRNA linked with an activated amino acid. Binding in the A site is greatly stimulated by the template polynucleotide. In this case, binding is codon specific. Binding of amino acyl tRNA is stimulated by the EFIA or EFTU protein. A site is formed by both ribosomal subunit. It is in the inter-unit space. In any case, the tRNA anticodon should be placed in the immediate vicinity of the mRNA codon, that is in the small ribosomal subunit where the acceptor end interact A site of PTC, that is with the large subunit. Then P site, peptidyl site or polymerization site, also known as donor site. P site is formed by both ribosomal subunit. It is in inter-subunit space, just like earlier mentioned A site. P site is formed by both ribosomal subunit is an inter-subunit space. It is the binding site for peptidyl tRNA. It binds the growing polypeptide chain of amino acids. At the same time, initiator tRNA aged binded to this site. It is the second binding site for tRNA in the ribosome. During translation, the P site holds the tRNA, which is linked to the growing polypeptide chain. It is this site that occupied by the peptidyl RNA prior to transpeptidation in the translating ribosome. It is vital that the peptidyl tRNA should not be exchangeable with the medium during translation. In contrast, when the deacylated tRNA or amino-acyl tRNA occupies the P site, the site becomes exchangeable and the tRNA may be released. An E site or exit site, it is the site for binding of the uncharged or unloaded tRNA. As a result of translocation, the deacylated tRNA is expelled from the P site. However, it doesn't necessarily mean that it is immediately released from the ribosome. The deacylated tRNA after translocation transiently retained on the ribosome in the exit site or E site. The E site, which binds the 3' terminus of deacylated tRNA in the vicinity of the PTC of the large subunit, seems to contribute mainly to the mention of. As a result of translocation, the deacylated tRNA is expelled from the P site. However, it doesn't necessarily means that it is immediately released from the ribosome. The deacylated tRNA after translocation transiently retained on the ribosome in the exit site. The E site, which binds the 3' terminus of the deacylated tRNA in the vicinity of the PTC of the large subunit, seems to contribute mainly to the retention of the deacylated tRNA after translocation. It is not clear that yet how other parts of the ribosome, especially a small subunit, are involved in the formation of E site. Then PTC, peptidyl transferase center. Peptidyl transferase center, catalysis of peptide bond formation. There is a peptidyl transferase center in the large subunit of ribosome, which contains two substrate binding sites, acceptor binding site and donor binding site, known as D and A sites of PTC of several peptidyl transferase activity, is the main and seemingly the only catalytic function of the ribosome itself. It is responsible for the formation of peptide bonds during polypeptide elongation. In the translating ribosome, transpeptidation proceeds between the peptidyl tRNA and the amino-acyl tRNA. In this reaction, the peptidyl tRNA serves as a donor substrate and the amino-acyl tRNA and an acceptor substrate. During transpeptidation, the PTC binds the 3' terminal adenosine with the amino-acyl residue of the A site bound tRNA at the site and the 3' terminal CCA sequence with amino-acyl residues and its peptide group of the P site bound tRNA at the D site. PTC is located at the large subunit on its interface site under the head and more exactly in the region of the groove separating the head from the rest of the body. There have been many attempts in isolating the enzyme from large subunit. It is responsible for catalyzing transpeptidation. However, none of these attempts has proved to be successful. It was concluded that the enzyme may consist of several proteins highly integrated in the large subunit that undergoes disruption in the course of protein isolation. Then it is estimated that L2, L3, L4, L6, L15 and L16 have found to be essential for peptidyl transferase activity. Then factor binding site will be there. GTP dependent binding of translation factors occurs here. It is located on the large subunit. Elongation involves the periodic binding and release of proteins EF1A, EF2, and EF2, EFG by the translating ribosome. Each of these proteins is bound in the complex with GTP and their release is in the result of GTP hydrolysis. The binding and release cycle of EF1A take place during amino-acyl tRNA binding, whereas the binding and release of EF2 proceeds during the translocation change. Also, initiation of translation involves the ribosomal binding of protein IF2 and GTP and the release of these proteins as a result of GTP hydrolysis. Finally, in the termination of translation, the ribosome binds and releases the RF protein. GTP takes part in this process as well. All these proteins interacting with the ribosome in the form of their GTP complex appear to bind to the same region of the ribosomal particle. It is likely that their binding sites in the ribosome are either identical or strongly overlapping. In any case, these proteins compete against each other for the binding site on the ribosome and cannot be present simultaneously. The two ribosomal subunits remain bound to each other during protein synthesis. The upper critical level of Mg++ ion concentration promote the association, and below critical level of Mg++ ion concentration promote dissociation of two subunits. Ribosome in protein synthesis. The ribosome carry amino acids to form particular proteins which are important to control the cell's activities. Protein is required several for cellular functions. The deoxyribonucleic acid makes mRNA by the step of DNA transcription. With the help of ribosomes, mRNA converted to protein. Processing of pre-rRNA and role of snRNAs. Processing involves the series of cleavage by endoribonuclease and exoribonuclease, which remove the external and internal transcribed spacer region, and base modification mostly by methylation of the 2' hydroxyl group of specific ribose and conversion of specific uridine residues to pseudouridine. By the time a human pre-rRNA precursor is first cleaved, over 100 methyl groups have been added to ribose groups in the molecule, and approximately 95 of its uridine residues have been enzymatically converted to pseudouridines. The functions of methyl groups and pseudouridines is that these modified nucleotides may protect the pre-rRNA from enzymatic cleavage, promote folding of rRNA into their final three-dimensional structure, and promote interaction of rRNA with other molecules. The processing of pre-rRNA is accomplished with the help of large number of small nucleolar RNA or snRNAs found in nucleolus. The snRNA function in association with specific protein and thus form ribonuclease proteins. There are about 80 different snRNAs in yeast and almost twice as many human chromosomes. These guide chemical modification of other RNAs, mainly ribosomal RNA transfer RNA, so they are also known as guide RNA. There are mainly two classes of snRNA. CD box snRNAs associated with methylation, HACA box snRNAs, CD box and HACA associated with pseudouridylation. The box CD snRNAs determine which nucleotide in the pre-rRNAs will have their ribosome moieties methylated, whereas members of other group called the box HACA snORNAs determine which uridines are converted to pseudouridines. Both groups of snRNAs contain relatively long stretches that are complementary to sections of the rRNA transcript. In this case, each snORNA bind to a specific portion of the pre-rRNA to form an RNA-RNA duplex. The bound snRNA then guide an enzyme, either a methylase or a pseudouridylase, within the snORNP to modify a particular nucleotide in the pre-rRNA. Taken together, there are 200 different snORNAs, one of each site in the pre-rRNA, that is ribose methylated or pseudourylated. If this gene encoding one of these snORNA is deleted, then action. Members of the box CD families have shared the sequence motifs given like this. Box C and box D located near the 5' and 3' ends respectively. Most CD snORNAs contain addition of C and D like motifs termed C and D dash, which are separated by a short spacer sequence. The CD snORNAs usually possess one or two long sequences that are complementary to rRNA. These sequences of 10 to 21 nucleotides are located just upstream from the box D, and D dash specify the site of 2 methylation in rRNA. The position complementary to the fifth nucleotide from box D gets methylated. So members of the box H ACA group are characterized by the sequence ANANNA box H by a highly conserved ACA triplet box ACE. Box ACE is always positioned 3 nucleotide upstream from the 3' end. The hairpin are generally interrupted by an internal loop, single-stranded bulges located close to their base. It contains sequences that are complementary to rRNA that undergoes base modification. The box HACA snRNA select the target rRNA sequences by forming 2 short helical nucleotides that are separated by 2 unpaired RNA nucleotides. The first unpaired base in rRNA sequence is a U residue that gets isomerized to pseudouridine. The pseudouridine has always position 14 to 60 nucleotide from box H or ACA. The SNO-RNP protein utilizes this structural information to select the correct target nucleotide. It is almost certain that the 2O-methyl transfer and uridine to pseudouridine isomerization reaction are catalyzed by fibrillary and discarin SNO-RNP protein respectively. Synthesis and processing of 5S RNA. The 5S RNA, about 120 nucleotide long, are encoded by a large number of identical genes that are separated from other rRNA genes and are located outside the nuclear organizer region. Following its synthesis, the 5S rRNA is transported to the nucleolus to join the other components involved in the assembly of ribosomal subunit. 5S rRNA gene is transcribed by RNA polymerase III. The gene for 5S rRNA contains an internal control region in its middle region, which is found essential for transcription. To this control region of gene remain attached a special protein called transcription factor 3A or TF3A, which permits RNA polymerase III enzyme to recognize the promoter of 5S rRNA gene. With only minor additional processing to remove nucleotide at the 3' end of the 5S rRNA diffuses to nucleolus, where it assembles with the pre-rRNA and remain associated with the region that is cleaved into the precursor of large subunit. Next is about biosynthesis of ribosomal proteins. Finally, all these rRNAs and protein assemble each other, forming large and small subunit separately within the nucleolus and then transported to cytoplasm. Location: nucleolus to cytoplasm. There is 2 types of protein that become associated with the RNA as it is processed. Ribosomal protein that remain in the subunit and accessory protein that have a transient interaction with the rRNA intermediate are required only for processing. And among the later group are more than a dozen RNA helicase, which are enzymes that unwind regions of double-stranded RNA that are necessary for the disload the SNO-RNPs. Then once the pre-40S particle leaves the nucleolus, it transfers the nucleoplasm quickly and is exported through nuclear pre-complex or N species. Once the pre-40S particle leaves the nucleolus, it transfers to the nucleoplasm quickly and is exported through nuclear pore complex or NPCs. The final step in the maturation of a small subunit occurs in the cytoplasm. In contrast to the pre-40S particle, the precursor of the large subunit requires considerable remodeling through many more transient interaction with non-ribosomal protein before it's sufficiently mature to export to the cytoplasm. So consequently, it takes longer time for maturing 60S subunit to exit the nucleus, 30 minutes compared with 5 minutes for export of the 40S subunit in filtered human cells. So multiple presumptive RNA helicases and small G proteins are associated with the maturing pre-60S subunit. Some RNA helicases are necessary to disload the SNO-RNPs. So once again, let's have a quick recap about ribosomes, its production, structure, composition, etc. So ribosomal large subunits is shown here. Amino acids are there. tRNA is there. mRNA present. Then ribosome small subunit is also there. So ribosomal large subunit is there. Amino acid, tRNA, mRNA, and ribosomal small subunit is also present. Then, let me just try to learn about So ribosome is an intracellular structure made up of both RNA and protein, and it is site of protein synthesis. The ribosome reads the messenger RNA sequence and translates that genetic code into a specified string of amino acids which grow into a long chain that fold into form protein. Ribosomes are discovered by George Emil Palade. He is a Romanian American cell biologist George Emil Palade using an electron microscope. Then structure. Ribosomes are compact ribonucleoprotein formed of ribosomal RNA and protein, composed of two subunits. One is twice the size of other, known as large subunit, and other one is called smaller subunit. The ribosomes are present in both eukaryotes and prokaryotes associated with some function. In prokaryotes, ribosomes are present in the cytoplasm as free, where in eukaryote it commonly associates with the membrane of ER or with cytoskeleton. In eukaryotic cells, their proportion of total protein is less, but the absolute number is greater. The rRNA provides most of the mass of the particle. In actively growing bacterium, there may be roughly 20,000 ribosomes per genome. They contain about 10% of total bacterial protein and account for approximately 80% of total mass of cellular RNA. Then prokaryotes, the small 30S subunit and large 50S subunit, while eukaryotes contain 40S subunit and 60S large subunit. And large subunit is dome-like shape. It carries the small subunit at its tip, forms a cup-like structure. In prokaryotes, the subunits are separated by a cleft. The two subunits are joined together by the concentration of Mg2 plus ions. When the concentration reduces, the two subunits will separate. Okay. Then polyribosomes. During protein synthesis, many ribosomes are aggregated by a common messenger RNA from a structure called polyribosomes or polysomes. So formed in the elongation phase, multiple ribosomes move along the coding region of mRNA producing a polysome. The ability of multiple ribosomes to function on a mRNA molecule explains the limited abundance of mRNA in the cell. Polysome activity can be used to measure the level of gene expression through a technique called polysomal profiling. Polysomes are shown. Ribosomal RNA. The 70S ribosome contains three types of rRNA. The 23S, 18S and 5S. The largest subunit 50S composed of 23S and 5S rRNA. Smaller subunit 30S formed by 16S rRNA. The nucleotide present in rRNA are given. The 80S contain four types of rRNA: 28S, 18S, 5S and 5.8S. Large 60S subunit contain 28S, 5S and 5.8S. The smaller subunit 40S contain only the 18S subunit type. Ribosomal protein. The 70S ribosomes of E. coli consist about 55 ribosomal proteins, and the 80S contain about 79 ribosome of proteins. These are proteins that helps to maintain the overall structure. Many proteins take part in the formation of active centers of ribosome, and in addition to the rRNA and protein, there is some metallic ions such as Mg2+, Ca2+, and Mn2+. In active centers, ribosomes have four major active centers formed by protein. A site, amino-acyl site, binds amino-acyl tRNA, mostly in 50S. P site, peptidyl site, binds peptidyl tRNA, mostly 30S. E site or exit site binds deacylated tRNA. E site, deacylated tRNA, mostly 50S. And mRNA binding site binds mRNA at 30S near to P. A site, amino-acyl site, P site, peptidyl site, and E site, exit site, binds deacylated tRNA, mostly 50S. 4 mRNA binding site binds mRNA at 30S near to P. And outgoing empty tRNAs, they are growing peptide chain, amino acid, incoming tRNA carrying amino acid, large subunit, mRNA, small subunit, etc. are there. And ribosomes and prokaryotes and eukaryotes, sedimentation coefficient, which is not an additive value, Prokaryotic 70S, eukaryotic 80S. Number of subunits 2, large one and small subunit. Same is the case with eukaryotes, 2 subunits. Large subunit 50S, large subunit 60S in case of eukaryotes. Then small subunit 30S in prokaryotes, but it's 40S in eukaryotes. So components of large subunit. 5S rRNA, 23S RNA, 34 different proteins. Components like this are given later. Models of ribosome structure. 2 models have been proposed to explain the structure and the association of 2 subunits, which are quasi-symmetrical model proposed by Stoffler and Wittmann. According to this model, the smaller 30S subunits have a bipartite structure with an elongated, slightly bent prolate shape. A transverse cleft divides the subunit into 2 parts of smaller head and larger body, giving the appearance of telephone receiver or embryo. And there are 3 protuberances in large subunit, central being the most prominent, giving the appearance of armchair. The next one is the asymmetrical model proposed by Lake. The 2 subunits are asymmetrical. The smaller subunit has a head, base, and so proposed by J. Lake. The smaller subunit has a head, base, and a platform. Base contains a cleft which separates the head and platform. Cleft is functionally important region where the codon-anticodon interaction taking place and part of binding site protein synthesis. Larger subunit contain 3 protuberances, central and 2 lateral called a stalk. The ridges are separated by the valley. The smaller subunit position over the large one and its head is aligned to the central projection of the larger one. Then reconstitution experiment of ribosomes. To understand the 3-dimensional organization of ribosomes and the interaction of rRNA and protein molecules, put forward by Nomura, purified the 30S subunit and dissociate them by chemical means into their components and allow them to reassociate under appropriate conditions. So ribosomes are centrifuged in gradient 5 mCsCl. About 30 to 40% of protein is lost. Then brain subunit are both the subunits are separated to 2 inactive core particles which contain rRNA and some protein. The dissociation generates 23S and 42S protein cores from the 30S and 50S respectively. The released protein are called split proteins. When this added to their corresponding core particle, the ribosome is reconstituted. There is 2 stages in reconstitution. The first stage is 30S subunit is to assemble the 16S rRNA with a group of 50 proteins. They react with the cold to form R1 particles, reconstitution intermediate. The second step, the R1 protein are heated in 48 degrees Celsius in the presence of other protein that are remained in the supernatant and forming an excited intermediate with in 20 minutes 30S is formed. The self-assembly of 30S is highly specific. In vitro reconstitution is a powerful tool for investigating the ribosome functions and biogenesis as well as the new features of ribosome.